So when we left off, I still scroll through this. Yeah. Okay, so we're talking about solute balance. So we're talking about how we maintain homeostasis, maintain the concentration of sodium and potassium in our plasma, which is really important because, as we hopefully remember, sodium and potassium are really important for the function of, for example, our neurons, other excitable cells, and they're also going to have an effect on water balance because solutes basically exert a pull on water. So our solute balance, which we're also going to be calling electrolytes, when you hear electrolytes, that's the same thing, exerts, an effect on water. Okay, so we looked at sodium balance specifically as is related to the hormone aldosterone. And we looked at what we call the RAAS, or the renin, angiotensin, aldosterone system. So we looked at basically this flow of angiotensin being produced by the liver, and then renin being produced by juxtaglomerular cells in the kidney, specifically granular cells in the afferent arteriole, which convert angiotensin to angiotensin 1. And then they run into some ACE enzymes in the capillary endothelial cells, convert to angiotensin 2, which tell our cells to secrete aldosterone, which ultimately is going to basically cause solute reabsorption. And we looked at a number of ways this kind of pathway works, because that's a number of different sort of steps. So one step we looked at was angiotensin 2 and the direct effects it's going to have on cells and on other parts of our cardiovascular system, other parts of our nervous system. So we saw that angiotensin 2, in addition to causing the body to secrete aldosterone so that we reabsorb more sodium, is also doing vasoconstriction. It's also causing ADH secretion. It's also causing thirst feelings. All of this in order to increase your mean arterial pressure. So it's really all a response to keep your blood pressure up. So it's all going to be a response to falling blood pressure. And that's going to really be true about basically everything in chapter 19 is about regulating blood pressure. That's why we track water reabsorption and why we track solute reabsorption, because these two things are basically controlling how much water comes back into the plasma in order to basically conserve the amount of pressure still in that plasma versus how much we're going to excrete if we want to lower our blood pressure. So that was kind of the point of this renin, angiotensin, aldosterone system. We looked at each of those pieces and how they would get, like why they would get released. So renin is kind of it's the first part of the RAAS, right? So anything that stimulates renin release, ultimately at the end of it is going to end up increasing aldosterone and therefore helping us reabsorb sodium, and we'll see reabsorb potassium as well, so that we can basically keep our plasma volume up by sucking that water through afterwards. So we saw that the stimuli for renin release are really all related to drops in blood pressure. But if we're thinking more directly about their effects and like how they're actually triggering those granular cells to release renin, we saw that there's decreased pressure in the afferent arteriole, which would be due to low blood pressure. We'd increase renal sympathetic nerve activity, which is basically part of our baroreceptor reflex, so also a response to low blood pressure, and decreases in sodium and chloride concentration in distal tubule filtrate, which is a response to low blood pressure, because low blood pressure means a low glomerular filtration rate, but because we have active pumping of these solutes into our medulla in order to keep the osmolarity of the medulla up, that lowers the concentration of those ions in filtrate. So that's another way we can basically sense that we have low blood pressure. Okay, so this is basically where we left off. So we'd finished talking about the system. which is basically a response to low blood pressure that causes us to reabsorb solutes in order to increase osmolarity, basically to pull more water along with it, ultimately. Now we're basically going to be talking about the flip side. So our next molecule that we're thinking about is atrial natriuretic peptide, or ANP. And ANP is going to get secreted by cells in the heart, so in the atria, in response to distension of the atrial wall, which is just to say that, like, when the atrium gets stretched, it's going to start releasing this peptide. Okay, so when this is basically a response to high blood pressure because you're responding to a lot more blood in the atrium. And so we're going to see that this is also related to increases in glomerular filtration rate. All right, so if we have a high blood pressure, a lot of blood flowing through, this is going to create dilation in the afferent arteriole. So that arteriole is going to get bigger. We're going to constrict the efferent arteriole. So basically what we're doing is like pushing flow really high into the glomerulus and then stopping it from getting back out. So we're doing this basically to increase the hydrostatic pressure in the glomerulus so that we can affect the solute concentration of urine, kind of. So our overall effect is going to be that we're going to be increasing the amount of sodium that we're excreting. So when we have high blood pressure, what we're going to try to do, like if we step back, is that we're going to try to get rid of more of these solutes and kind of lower the solute concentration in plasma. And the reason for that is that when we lower the concentration of these solutes, it basically means that we're not going to reabsorb as much water. So that's kind of the point of this. And this is how it's going to sort of fix these blood pressure issues. So the way it's going to directly do that is by closing sodium channels in the apical membranes. So it's kind of doing the opposite thing to what aldosterone was doing. So aldosterone, we're trying to keep more sodium in the plasma because we want more water in the plasma. We want more water being reabsorbed because we want to keep blood pressure up because it's all a response to low blood pressure. So with ANP, we're responding to high blood pressure. So we're really trying hard not to increase salt content in the blood because we don't want more water in the blood. We don't want more plasma because that would just make our problem worse, basically. So that's part of why you see when you're thinking about sort of health problems, right? Like people who have high blood pressure, right, are told to decrease how much salt they eat, right? So this is kind of why it's related. So this is a hormone that basically is trying to do its job to stop your plasma from having too much sodium in it. Another way you can control that, right, is by consuming less sodium. So that's how those kind of like behavioral things go together with this physiology. So we can see in a flow chart basically how this all works. So if we register high plasma volume, and we remember that we have basically stretch receptors, right, that sense blood pressure because they sense stretch in the cardiovascular system. So we can sense that we have high plasma volume. And one of the places this is going to have an effect is in the atria. So the atria are going to basically get stretched out when there's a lot of blood moving into them because of high blood pressure. And the atria themselves, so the heart itself, secretes ANP. And then ANP travels through the cardiovascular system, right? So that blood in the atria goes into the ventricles, goes out through your arteries, and eventually circulates around to your kidneys. And in the kidneys is where it's going to have its effects that try to balance out that high blood pressure, try to fix that high plasma volume. And so they do that One way, by really increasing the glomerular capillary pressure. So they're increasing glomerular filtration rate, because everything that's in the plasma initially gets filtered out at the glomerulus. So everything except for our red blood cells and our proteins come out of the glomerulus. So if you have a higher glomerular filtration rate, it means you're getting potentially more plasma out of the body, right? Like everything that was in that plasma is going into our nephrons. So that's kind of a first step to getting rid of your sodium. You have to get it into the nephron. So we do that by pumping as much blood and therefore as much plasma into the glomerulus as we can by dilating the afferent arteriole, and then kind of getting it stuck there by constricting the efferent arteriole. So we're building up pressure in that capillary, and that pressure affects our balance of Starling forces, right? So one of the factors that increases filtration rate is the glomerular capillary pressure. So this is how we're upregulating glomerular filtration. Okay. So the other thing that ANP is doing is it's decreasing sodium reabsorption by closing those sodium channels in the apical membrane of our principal cells. So we have a lot of sodium flowing through the nephron just because we've increased the glomerular filtration rate. So we have a lot of everything flowing through the nephron, but then we're at the same time stopping it from getting reabsorbed. So if we have a lot in there, we stop it from getting reabsorbed, it has nowhere to go, so it gets excreted. So you're gonna end up with a bunch of sodium in the urine, which means that you don't have the sodium left in your plasma to pull, to basically pull water towards it. So another thing that's happening at the same time as this, which isn't, we'll get to how all these things play together actually before I say it isn't related to ANP, because that's not strictly true. But a thing that's happening at the same time, right, is if you had a lot of renin being produced, and we're starting out that RAAS process, right, to secrete aldosterone, you'd be fighting against yourself. So at the same time as ANP is increasing our glomerular filtration pressure and stopping sodium from being reabsorbed, we're also going to want to decrease renin secretion so that we cut off that renin, angiotensin, aldosterone system at its beginning, right? Like we want to nip it in the bud. So we're going to decrease renin secretion so that we decrease aldosterone, so that we also have the effect of decreasing sodium reabsorption, because those are the two things that are like enabling us to reabsorb sodium. Okay. So we will talk about these later. Maybe we'll go through like all of these questions on our review day or something. I think that would be a good thing to do. Also maybe build a big flow chart together as a class about like blood pressure and how it affects all of these different hormones, because they all kind of have effects that work together. I got a question? No, just leaning on the space bar. Okay. So we've looked already at how aldosterone affects the sodium balance, but it's also affecting potassium balance. So we're back to aldosterone. Took a detour to ANP, now back to aldosterone. So another thing that we're trying to control is potassium, because we can also have bad effects if you have too much or not enough potassium, because remember that potassium, right, is critical to how our neurons work, because it's basically pulling our membrane potential back up, right? It's the second part of how an action potential propagates, right? We have to get our membrane potential back to normal. Okay. So hyperkalemia, high plasma potassium. Hypokalemia is low plasma potassium. And aldosterone is one of the things that's going to help us control the amount of potassium, just like it's helping us control the amount of sodium. And that's process is going to look similar, but there are going to be a couple differences. So since we're now talking about potassium, let's just talk through what's happening with potassium at each step of the nephron. Basically going to be similar to what's happening to our other molecules, but we're following just the potassium ion to make sure we got it straight. So at the glomerulus, it's getting freely filtered out. So it's one of the things that's in the plasma that can just go through those membranes and get into Bowman's capsule. So we're just pushing potassium out as we push out sodium, as we push out water, as we push out the other contents of our plasma. In the proximal tubules, just like we see sodium reabsorption, just like we see water reabsorption, we're also going to see potassium reabsorption. As we get to the distal tubules and the collecting ducts, here's where we're going to see a little bit of a difference. So potassium is going to be both reabsorbed and secreted. So part of what's going to influence our potassium balance is that secretion part. So potassium is going to be pushed back into the tubular fluid in the distal tubules and the collecting ducts. So when we're thinking about our potassium balance, we have to factor in both the potassium that's getting moved out of the fluid in the tubules back into the plasma and the potassium that's getting removed from the plasma again and secreted back into the tubular fluid. So that's like part of the point of that equation we looked at way back, might even have been 2 weeks ago, about how we can tell whether something's being reabsorbed or secreted based on the amount that's getting filtered out and the amount that's getting excreted. One of the things we might want to track that way is potassium, because this is one of the places where secretion plays an important role. Okay, so our potassium secretion in the distal tubules and the collecting ducts is going to be regulated. So that's under active control. It's not just happening automatically, and it's going to be happening through the action of aldosterone. So aldosterone, as relates to potassium, like potassium in the plasma is going to directly stimulate aldosterone release. So when we were talking about sodium balance, we were thinking about renin, angiotensin, angiotensin 1, angiotensin 2, all the way down to aldosterone. We're going to see that aldosterone is also just immediately secreted when we sense an increase in potassium in the plasma. So as we increase potassium, we're going to release more and more aldosterone. So that's kind of a direct relationship. Okay. So now we're going to look at some pictures of what the potassium is doing in our different types of cells. So here we see our proximal tubule principal cell. So our basolateral membrane, our blood would be over here, off to the right. We got our apical membrane facing the lumen of our proximal tubule. So here's where our tubular fluid will be. We've talked before, we've got our sodium-potassium pumps on the basolateral membrane. They're always there. One of the things aldosterone is doing is increasing those, right? We also have in that basolateral membrane channels for potassium. So it basically leaks potassium back out, right? We actively pump potassium into the cell, but then it can move down a concentration gradient back out of the cell. This is kind of a loop. Another thing that does happen, although they actually don't know exactly what kind of transporter is doing this, we have some type of membrane protein that's pushing potassium from the tubular fluid into epithelial cells in the proximal tubule. So that's an avenue of current research. So, but we'd expect it would probably be some type of active transport because we have our sodium-potassium pumps keeping potassium concentration kind of high in the epithelial cells, which is why it can move passively down its gradient through these sort of like leak channels. So we're going to have some kind of active transport potassium to get potassium into the cell from the apical side. Another thing that's happening is through these leaky tight junctions, we have potassium moving paracellularly, so between epithelial cells. We have potassium moving directly from the tubular fluid, where it's potentially kind of high, right, into the paratubular fluid. Did I say? I'm not sure I said that right. Moving from the tubular fluid into the paratubular fluid. Our potassium concentration in the paratubular fluid, so the part between the epithelial cells and our capillaries, is low because of the action of that sodium-potassium pump in the basolateral membrane. Okay. So that's the reabsorption that's happening at the beginning of our nephron, but this isn't really where we're going to see the effects of aldosterone. This isn't really the control part. Okay, so we're going to move next to our distal tubule, so the late distal tubule, which is the part dumping into the collecting duct, so then also into the collecting duct. So what we have happening there at the end of our nephron, still our same principal cell, we got our tubular fluid on one side, paratubular fluid on the other side. We still have our sodium-potassium pump on the basolateral membrane, but what we also have is a potassium channel on the apical membrane. Okay, so I'm going to flip back for a second just to point out that when we're looking at our proximal tubule, there are no potassium channels here. So our potassium was getting reabsorbed, but it's not moving back into the tubular fluid, even though the concentration in this epithelial cell is kind of high because of the action of the pump. But in the late distal tubule and in the collecting duct, because we have these channels in the apical membrane, we have that same situation if we're looking at the potassium concentration. So low potassium concentration in the paratubular fluid, high potassium concentration in the epithelial cell. But now that we have a channel in the apical membrane of our principal cell in the distal tubule or collecting duct, potassium is able to move down its concentration gradient from the epithelial cell into the tubular fluid. So this diagram is actually showing us secretion. So the secretion here is still happening on the apical membrane. It's happening passively. It's being actively sort of set up by our sodium-potassium pump, like almost everything else. But the secretion is going to be dependent on these potassium channels and whether they're open or not. So if we don't have potassium channels, that potassium is just going to get stuck in the principal cell. If we do have potassium channels and they're open, then we're going to see potassium being secreted back into the tubular fluid. So at this point, the potassium concentration of the tubular fluid is kind of low because we've reabsorbed potassium in the proximal part of the nephron actively. Right, so we were reabsorbing it actively here across the apical membrane through some mystery mechanism and also through these leaky channels because we're keeping low potassium concentration in the paratubular fluid because of our pump. Okay, so we reabsorbed our potassium that was in the tubular fluid at the beginning, so our potassium is low here. So potassium can flow down its gradient back into the tubular fluid if these channels are open. So unsurprisingly, the effect of aldosterone is to open channels. So it's opening potassium channels in addition to opening the sodium channels. You saw this slide before. I think I duplicated it and pulled it here just so that we could look at it again. So what we're seeing here, right, is our aldosterone in the plasma diffusing across our cellular membranes, getting in, touching that cytosolic receptor, and then having its effects. And so here, all it's doing on the apical membrane is opening channels, whether we're talking about sodium or potassium. But because the gradient of sodium and potassium are opposite to each other, opening a channel for sodium has the effect of reabsorbing sodium or pulling it out of the tubular fluid and into the plasma. But opening up a potassium channel has the opposite effect. So opening up a potassium channel causes secretion because that sodium-potassium pump on the basolateral membrane basically makes sure that sodium and potassium have the opposite concentrations of each other. So it keeps our sodium concentration in the cell low, but it keeps our potassium concentration in the cell high. Okay. So in our distal tubule and in our collecting duct, overall, if we're just thinking about what's getting reabsorbed and what's getting secreted, we're going to expect sodium will be reabsorbed, the potassium will be secreted, and aldosterone is going to control like how much of each of those is going to happen. Okay. Questions about potassium or ANP? We're going to skip the section on calcium balance, but calcium is also balanced by similar kinds of systems. It's important to balance calcium because it's important for basically bone growth, but also remember that calcium is important for releasing neurotransmitters, so functions of neurons. So we'd see different hormones basically that are affecting how calcium is moved in and out of the cells, but it would be the same kind of deal. Check the chat before we move on to acid base. Okay, so we're going to start with a little bit of acid base. I don't want to overwhelm us with that, but we got plenty of time right now. Okay. Sure, I'm sharing my thing. Acid base, cool. Okay. So when we're thinking about acid-base balance in the blood or acid-base homeostasis, we're actually going to be linking together the respiratory system and the urinary system. So our lungs are kind of related to our kidneys. So we're going to start by kind of going over the respiratory stuff. So that's actually not in chapter 19. I think that's the final section of chapter 17, which we didn't really talk much about. So we're going to talk about it now before we get into how the kidneys are working together with this system. Okay. So when we're talking about acid-base homeostasis, what we're really talking about is controlling the pH of the blood, which is maybe not surprisingly to you, but kind of surprisingly to me, really important to how your body functions, because it's basically going to maintain appropriate protein function. So we're going to look at a brief review of pH, the pH scale, what pH means, and then we're going to talk about how the acid-base balance can get disturbed in the blood, so how blood can become too acidic or too basic, too alkaline. And we're going to think first about the role of the respiratory system in that balance before we get to the kidneys. Okay. So the pH scale goes from 0 to 14. It's going to be regulated by our respiratory system and our renal system. We're talking about our renal system now. We're going to link in the bits from the respiratory system before we move into the direct actions of the kidney. And actually, very small changes can have large physiological effects in the body. So a normal pH range for your blood and is like 7.35 to 7.45-ish. I think it's actually supposed to be 7.42. That's our normal range. So we're actually, not right at 7. We're not right in the middle. We want to be a little bit above. We can have conditions that create acidosis. So if we see an increase in the hydrogen ions in the blood, we're going to lower our pH and become more acidic. If we decrease the amount of hydrogen ions in the blood, we're going to see conditions that create a condition of alkalosis. So our pH can also get too high. And both of these are basically a problem because of how proteins work. So way back when in ANP1, we talked about protein folding and how proteins have a shape due to interactions between the different amino acids that make them up, right? So we had our 20 amino acids. They all have different chemical properties. So they have different charges in different places. And they're basically the attractions between them cause your protein to be some shape. And it's actually really hard to predict what shape a protein will be. But the shape of that protein is critical to how it functions, right? So we thought about that most when we were thinking about enzymes, but it's true for all our proteins. So remember, with an enzyme, we have to kind of like fit our substrate into the enzyme in order to create bonds or to break bonds. If we have the wrong pH in the blood, suddenly our proteins are going to, I don't want to say fold into the wrong shape, but like if you have extra hydrogen ions or too few hydrogen ions, which is what a different pH is, those are charges, right? So those are different charges that are going to interact with the proteins. We saw that hydrogen bonds are also really important for protein structure and protein shape. So if you change the pH of the blood, the relationships between those amino acids is going to kind of shift, which is going to change the shape of a protein. And if you change the shape of a protein even really slightly, it was supposed to match up, right, to some substrate and catalyze some important chemical reaction, you're going to totally mess up that chemical reaction. And that can be fatal. It can be fatal within seconds, actually, depending on how much that pH is disturbed. So we really want our proteins to function normally, and proteins are doing practically everything in our body, right? Because it's not just the enzymes, it's also, right, those channels that we were just looking at, those are proteins. Our sodium potassium pump is a protein, right? And if and imagine if you turned off all the sodium potassium pumps in the body, this whole kidney function would definitely be screwed, but so would a bunch of other things, right? Our neurons are totally dependent on those voltage-gated channels in order to send their action potentials. If you mess with proteins, you mess with basically everything. Okay. So there are some things you can observe, basically in people or in the nervous system, that are indicative of either acidosis or alkalosis, so a pH that's too low or too high. They have kind of opposite effects. So if we have acidosis, so our pH is too acidic, this actually depresses the central nervous system. So partially, right, by messing with membrane potentials, right? So it's going to be harder to fire off action potentials, which means your nerves are going to fire less, which is a problem. In alkalosis, we see the opposite. So it actually creates a hyperactive nervous system, which is a problem because that causes things like seizures. So both of these are a problem. In acidosis, you might end up in a coma. In alkalosis, you might end up having seizures all over the place. Big problem. Okay. So I put the textbook section for the respiratory system part of the acid-base balance. So go back to chapter 17 to review this, or really to get details on the respiratory system part. It's in 17.8, but we're going to talk about it now. So one of the things that's controlling our acid-base balance in the blood is hemoglobin. So hemoglobin in our red blood cells, right? Remember, it's those 4 globin proteins and then our heme group. In addition to binding oxygen and carrying oxygen around, hemoglobin binds and releases hydrogen ions. So hemoglobin bound to oxygen can be converted to a molecule of oxygen, a molecule of hemoglobin, and a molecule and a hydrogen ion. And then that can be converted basically to hemoglobin and hydrogen, right? If we get rid of the oxygen. So we can have hemoglobin bound to hydrogen or hemoglobin bound to oxygen. specifically deoxyhemoglobin, so hemoglobin without that oxygen bound into it, binds hydrogen ions better. So that's why it's drawn out like this with hydrogen bound on one side, oxygen bound on the other side, is that when we don't have oxygen bound to our hemoglobin molecules, or our hemoglobin units, we're going to end up binding on more hydrogen ions. So we're going to basically soak up more hydrogen ions from the blood. Okay, so this means, oh, well, actually, we'll link these together in a second. Another thing that's going to work as a buffer are bicarbonate ions. So bicarbonate ions act kind of like a chemical sponge. So our bicarbonate ion here, actually let's start from the other side. So our bicarbonate ions are ultimately going to be coming from CO2 in the blood. So if we take our carbon dioxide and water, we can make carbonic acid, and then carbonic acid dissociates into bicarbonate and hydrogen. So our bicarbonate ions can also do the reverse. All of these reactions are reversible. So bicarbonate ions can bind a hydrogen ion, become carbonic acid, or carbonic acid can dissociate and let go of those hydrogen ions. So our bicarbonate ions, therefore, can kind of control the pH of our blood, because they can either pull those hydrogen ions out of the blood or let them into the blood. And this is related to the respiratory system, because the way that we get carbonic acid, which can dissociate into bicarbonate and hydrogen ions, is from CO2. So we consider CO2 a respiratory acid for this reason, because if we dissociate it all the way, we're going to let go of these hydrogen ions. So normally we have a specific balance between carbonic acid and bicarbonate. So we're going to have this basically 1 to 20 relationship, and we're going to extend that to a specific relationship between CO2 and bicarbonate as well. But you can see that here, when we have this balance, we're going to be at our normal pH, which is between 7.35 and 7.45. If we move it to the left, right, so if we increase our amount of carbonic acid, we're going to end up in acidosis. If we get below about 6.9, that's deadly. In the reverse, if we increase our bicarbonate, we're going to end up in alkalosis. Eventually, this will also cause death. Okay. There is an equation that tells us the relationship between bicarbonate, carbon dioxide, and pH. It's called the Henderson-Hasselbalch equation. So here's the equation written out: 6.1 plus log bicarbonate over carbon dioxide is the pH of our blood. Okay, so this basically means that if we increase CO2, we're going to end up with a more acidic pH, or if we decrease CO2, we're going to end up with more alkaline pH. So we want to maintain our normal arterial pH at 7.4. And in order to do that, we have to have this very specific ratio to make this equation come out to equal 7.4. And that relationship is 20 bicarbonate to 1 molecule of CO2. So the respiratory system is the part that's going to be regulating the CO2. So that's why the respiratory system has to play together with our renal system, because we have to balance both the CO2 and bicarbonate in order to keep this pH steady. So the respiratory system is controlling how much CO2 you have in your blood, basically by controlling, right, how much CO2 you're giving off, how much CO2 is being returned to your alveoli, how much is being removed through ventilation. And then your kidneys are also able to assist in this balance. Like, say you're not breathing out enough CO2 or something, your kidneys can help regulate pH by regulating the bicarbonate ions, which is important because you wouldn't want to have holding your breath or something kill you by messing with your blood pH. So it's important that you can do metabolic regulation of blood pH as well. Because there are also other sort of even longer term things that can change the respiratory side of this equation. Like if you're at a different altitude, that affects concentration of O2 and CO2 in your blood. And so you want to be able to kind of adjust for situations like that. Okay. So here written out, the 2 disturbances we're going to want to be on the alert for. So if we have an increase in CO2, so you're not breathing out enough that's staying in your bloodstream, you're going to end up with acidosis. So if this acidosis is, the reason it says respiratory acidosis is we'll see that there's also acidosis and alkalosis that can be caused by the renal side of the equation. But respiratory acidosis means that you have a lot of CO2 in your cardiovascular system because you're not breathing out enough CO2. So that's a problem. That's a disturbance to our acid-base balance. And the reverse is also a problem. So if you have decreased CO2, you're going to have higher pH, and that's respiratory alkalosis. So we are going to stop after this section, but I just want to pull up one of the previous slides with the equation because I think it can be kind of helpful. Okay. So like this one, right? So like if you increase the amount of CO2 here, right? So you're increasing the carbon dioxide in your blood. That means you're going to kind of force the equation this way, right? So you have increased CO2, so you're going to create more carbonic acid. When you create more carbonic acid, you're basically going to release a bunch of hydrogen ions. And if you decrease carbon dioxide, it forces the equation kind of in the other way. So it's going to decrease carbonic acid, which means that your bicarbonate is going to suck up those hydrogen ions. So that's why we basically see these effects of increasing CO2 and decreasing CO2 respectively causing acidosis or alkalosis, is it's just driving these reversible reactions in specific directions. So I find it a little bit easier to kind of just like remember this relationship in terms of the equations instead of just kind of memorizing which does acidosis or alkalosis, but your mileage may vary on that. Okay, so we're gonna stop here. So questions on the respiratory side of acid-base balance. Check here. Okay, so I will make sure that these videos are posted rapidly. Sorry guys that I've been a little slow on getting these on YouTube. I've been trying to edit them because stuff keeps going wrong with the video, but my little laptop cannot really handle rendering these videos. But I promise I will have everything up by the end of today. All right, so that's it for now.